from marine natural product to drug. Natural products and compounds derived from natural products have always been used in the treatment of human diseases. The success rate for natural product to be developed into drug is 0.3%, and it is 300 times higher than in the case of synthetic compounds. Among these, marine natural compounds show higher incidence of significant bioactivity, which is associated with a rare and unique chemical structure. However, developing a new drug from natural product is a great challenge, and it requires an interdisciplinary approach. This long-term process usually takes 20 to 30 years, and it includes basic research, preclinical and clinical trials. Finally, the new drug has to be approved. Searching for new bioactive natural products starts with biomass extraction. Then the crude extract is fractionated and the obtained fractions are screened for biological activity. The sample that shows promising activity but still contains a complex mixture of compounds is subjected to further bioassay guided fractionation. Finally, the pure bioactive agent, called heat, is isolated and characterized. In this process, the automated high-throughput screening is used to select the new chemical entity, NCE, with the best pharmaceutical properties. Samples are tested against biological targets such as enzymes, cancer cell lines, or cell surface receptors. The results of screening experiments supply important directions for lead optimization. As a result, agents with better therapeutic index, it is with superior activity and decreased toxicity, are obtained. Before testing a drug candidate in humans, information about its safety and efficacy and the potential to cause serious harm effects should be available. To collect these data, in vitro, in vivo, and in silico, preclinical tests have to be done. In vitro tests use microorganisms, cells and tissue cultures, subcellular components, or biomolecules such as enzymes. In vivo tests are performed on animal models, rodents and non-rodents. In silico tests are based on computer simulations. They, among others, help to predict the receptor binding site for the tested compound. The in vitro and in vivo tests include studies on adsorption, distribution, metabolism, excretion, and toxicity of drug candidate, so-called ADMET. The tests are critical for establishing safety dose range and administration schedule. Finally, natural product as investigational new drug, IND, with promising outcomes in preclinical studies, proceeds to clinical human trials. The trials are done in 4 phases with increasing number of individuals. If the results of the first 3 phases of the trials are positive, the drug can be approved by the American Food and Drug Administration Agency and by European Agency for Evaluation of Medicinal Products. After the approval by these regulatory agencies, long post-marketing studies are conducted. The aim of phase 4 of clinical trials is to control the safety and efficacy of the product in real world. Even at this stage, the drug can be withdrawn from the market. Drug development from marine natural resources is usually hampered by such difficulties as sustainable source and structure complexity of biomolecule. Therefore, alternative strategies such as chemical synthesis are being developed.